Your plant is dissolving. This is not a problem. This is not a deficiency. This is not a sign that you have done anything wrong. This is, in fact, a plant doing something quite sophisticated, and it is doing it on purpose. What you are watching is a controlled demolition, and the contractor is the plant itself. Almost every plant you put in this tank came from somewhere above the waterline. A farm, a greenhouse, a holding tank with the water level kept low. It was growing in air, producing leaves designed for air. Thick-walled, waxy-surfaced, built to resist desiccation and manage gas exchange through stomata on the leaf underside. Those are emersed leaves. And emersed leaves are, from the plant's perspective, the correct leaves for the environment the plant was in. They are not wrong, they are just no longer appropriate. Submerge that plant, and the environment has changed in almost every measurable way. The light quality is different. It is now filtered, sifted, attenuated by the water. The carbon dioxide is dissolved rather than gaseous. And there is no desiccation risk. There is no need for a waxy cuticle. There is no need for stomata at all in many cases, because the gas exchange can happen directly through the leaf surface. The emersed leaf is overbuilt for this environment, and the plant knows it. The submersed leaf that will replace it is thinner, optimized for light capture in water rather than radiation management in the air. Often it is more elongated, often more translucent, because the plant underwater does not need to worry about overheating. It is a different solution to a different set of problems built by the same genome. This phenomena, one that the plant is producing morphologically distinct leaves in response to environmental conditions, has a name: Heterophylly. From the Greek, different leaves. Scientists, as previously noted on this channel, tend to describe what they see. Here's what the melt actually is. The plant has detected that it is submerged. It has received the signals: altered light spectrum, dissolved CO2, hydrostatic pressure, ethylene produced by its own submerged tissue. It has made a developmental decision. It is going to dismantle the emersed leaves, not passively, not because they are rotting, actively, deliberately, using a molecular process called apoptosis. Programmed cell death. The plant is killing its own tissue on a schedule, cell by cell, and in order that maximizes what it can recover before the leaf goes. That yellowing you see is not chlorophyll degradation from stress, it is chlorophyll withdrawal. The plant is pulling the photosynthetic machinery out of the leaf, disassembling it, and moving the components, the nitrogen, the magnesium, the phosphorus, back into the stem, where they will be used to build submersed leaves growing below. The leaf that looks like it is dying is, in a meaningful sense, being harvested. The plant is not losing resources, it is relocating them. When the plant finally detaches the leaf, what falls is mostly structural. Cell walls, vascular tissue, things the plant couldn't efficiently reclaim. The rest has already been accounted for. And that structural debris settles onto the substrate, where the decomposer food web we covered in previous episodes will take it from there. The melt is not the plant failing. It is the plant being extraordinarily efficient with a body plan that it no longer needs. Ludwigia. The emersed leaves are noticeably thicker. You can see it in the way the light moves through them compared to the new growth below. The color often shifts too. The emersed form tends towards deeper green or reddish brown depending on the light, while the submersed leaves, once established, will often show more of a red pigmentation the genus is known for, given sufficient light and nutrients, of course. Two different color strategies, same plant. Hygrophila. Among the most dramatic heterophylls in the hobby. The emersed leaves are broad, stiff, and have a texture that reads almost as substantial in the hand. The submersed form that emerges below can be noticeably narrower and softer, built for water, not for weight bearing in air. Some species in this genus show such extreme dimorphism that the two leaf forms look like different plants entirely. They are not. Bacopa, the most visually obvious transformation of the three. The emersed leaves are thick, almost succulent in texture, rounded and waxy, a plant that evolved in conditions where water availability was variable and desiccation was a genuine threat. Submerge it, and the new growth that comes is thinner, more translucent, adapted to the opposite problem, not conserving water, but capturing light through it. 3 genera, 3 different solutions to the same problem, 1 tank. All of them simultaneously building a new body plan for an environment that is new to them, but not unknown, because the genome has been solving this problem in one form or another for a very long time. What is happening in this tank has a name that extends well beyond botany: phenotypic plasticity. The capacity of a single genome to produce different phenotypes in response to environmental conditions. Same DNA, different body, depending on where that DNA finds itself. In a previous episode, we watched Daphnia dubia switch from clonal reproduction to sexual reproduction. The same genome, completely different reproductive strategy, triggered by environmental signals. A plant doing this with its own leaves is the same principle, expressed in a different kingdom across a different timescale. Phenotypic plasticity is not a workaround. It is not the organism making do. It is a sophisticated capacity for environmental reading and developmental response, evolved because environments change and fixed body plans are a liability. The plants in this tank are not adapting. They are doing exactly what they were built to do. Every discipline in biology has a name for this. Developmental biology calls it plasticity. Ecology calls it acclimation. The tank calls it Tuesday. Normal transition melt looks like this. The emersed leaves yellow and soften, beginning at the tips and the margins. The tissue becomes translucent. It is not slimy, it does not smell, it detaches cleanly. And critically, below it, new growth is actively emerging. The plant is trading up. That new submersed growth is the confirmation signal. If you see melt happening above and new leaves unfurling below, the plant is in transition, and it is proceeding correctly. You do not need to intervene. You should not intervene. You should, ideally, watch. Abnormal melt looks different. If leaves are rotting rather than yellowing, if they are brown, slimy, with a distinct odor, that is bacterial infection, not transition. If the melt is progressing faster than the new growth is appearing, and the growing tip itself is dying, something else is wrong. Perhaps there is lower light, nutrient deficiencies, or temperature shock. The diagnostic question is always the same: is there new growth? If yes, the plant is winning. If yellowing is spreading not from the emersed leaves, but from the oldest established submersed leaves upward, you may have a nitrogen deficiency. The plant is pulling nitrogen from old tissue to fund new growth, which means there is not enough in the water column. That is a different signal, a different problem, and a different solution. Every behavior is data. Plants are always talking. So, you have planted emersed-grown stem plants. They are melting. And you've read the signals and confirmed this is normal transition melt. What do you do? First, do nothing. It's actually quite fine. If you leave it, it will eventually fall off and decompose and feed the substrate biology. Or, if you must, once the leaf is fully gone and detached, you can remove it. Either way, the decomposers will find it. Second, do not uproot the plants to check the roots. Do not move them to a different spot. Do not add fertilizer in a panic. The plant is executing a developmental program that took millions of years to evolve. It does not need you to improve on it mid-execution. Third, Watch the growing tip. That is where the new body plan is being built. If the tip is healthy and the new leaves are forming, the transition is on track. The rest of the plant is just doing paperwork. Fourth, wait. That is the whole instruction. The plant knows what it is doing. It has been doing it longer than we have been watching. Your plant is dissolving. It is dissolving because it was built for a world above the waterline, and it has been asked to live in a world below it, and it is dismantling the first body to build the second. Systematically, efficiently, cell by cell, recovering what it can and releasing what it cannot, following a developmental program encoded in its genome before your tank existed. Ludwigia, Hygrophila, Bacopa, 3 genera, each navigating the same transition on their own schedule, at their own pace, by their own molecular logic. 3 different solutions to Heterophylly. 3 different versions of the same ancient decision. This environment has changed, and so must I. It is not a problem to solve, it is a process to observe. And in a tank built around the principle that every organism is a data point, a plant tells you, in the language of its own cellular biology, exactly what it is doing and why. It is precisely the kind of organism this channel was built to understand.